obtained his MBBS from Kakatiya Medical College and his MD from Gandhi Medical College. He has various research publications to his credit in reputed journals. We are truly fortunate to have him here with us to share his wealth of knowledge and insights. On behalf of Dr. SMCSA Medical College, Department of Biochemistry, and all students, I extend a heartfelt gratitude to Dr. Rafi sir for accepting our invitation. We look forward to an inspiring and thought-provoking session. So before starting the session, I would like to invite our principal, Dr. Arsha Merlin ma'am, to say a few words.
Good, Mary.
Most respected Dean, Dr. Rafi, HOD of our Biochemistry Department, Dr. Vijata Thomas, faculty members of Biochemistry Department, and my dear students of 2024 MBBS batch. A very warm good morning to one and all. It is a real honor for us, sir, to have you with us in this virtual platform. So technology has advanced in such a way that even at a remote place, we are able to, and we are fortunate enough, we and as faculty as well as our students are fortunate enough to listen to the speech of eminent speakers like Dr. Rafi. I know being the dean, how busy your schedule would have been, and I could understand the passion you have for teaching our undergraduate students. I would like to say we are fortunate and our students are fortunate to have you with us, sir, in this platform. And on behalf of our institution, Dr. Somerville Memorial CSN Medical College and Hospital, Karakonam, I extend a very, very hearty welcome to you. And I am indeed proud and thankful to you for having you with us. And I am very sure that our students will have a virtual treat with your eminent speech, which will be a motivation and inspiration to all of us. Thank you very much, and I take this opportunity to congratulate the Biochemistry Department under the leadership of Dr. Vijata Thomas. Over to you, sir.
Thank you, madam.
So, as ma'am said, we are fortunate enough to have you here, sir. So, I would like to, tell the students, you all are fortunate. We are fortunate to have sir here with us. So, thank you for accepting the invite, sir. And thank you so much, Shanusha ma'am, for, coming here in spite of your busy schedule to make sure you are here. Thank you so much. I would like to introduce you to our department faculty, and then we'll start the session.
Sure.
Hello, sir. I am Dr. Sajda, Associate Professor at Biochemistry Department.
Good morning, sir. I'm Dr. Soumya, Associate Professor in Biochemistry Department.
Hello, sir. Myself, Dr. Gracia, Assistant Professor in the Department of Biochemistry.
Good morning, sir. I'm Dr. Vishwanath. I am tutor of Biochemistry Department.
So I think we can start the session. So I'll stop sharing. Stop sharing. So you can start sharing.
Yeah. Can you hear me?
Yeah, we can hear you, sir.
Yeah. Shall we begin or?
Yes, we'll begin, sir.
Yeah. Good morning, students. So, very good morning. The principal, the respected principal, the faculty of Department of Biochemistry, the students of 2K24 batch of Somerville Memorial CSI Medical College, Karakonam, Kerala. It gives me immense pleasure to meet you all virtually, if not physically, to interact with you for a couple of hours. Hope if you can hear me properly, please raise your hands. Yeah, thank you. So my advanced greetings on the eve of Christmas to all the students and faculty. In fact, we celebrated yesterday evening the pre-Christmas party in our campus, where our students of 2K24 have taken part actively. So I take this occasion to greet one and all. Shall we begin our session? Yeah, thank you. Metabolism. Metabolism is finely modulated as per the needs of an organism. This fine modulation, this fine regulation is achieved by regulating the activities of the so-called key enzymes of these metabolic pathways. You know pretty well that all the metabolic pathways, all the major metabolic pathways are regulated by one or more key enzymes. So we call these key enzymes as regulatory enzymes, or more precisely, rate limiting enzymes. By regulating the activities of these so-called rate limiting enzymes, we will be able to finely modulate the metabolic pathways. So for this purpose, the body adopts to various strategies that we will discuss. So I'll try to apply these regulatory strategies of enzymes, which you might have already learned during learning about enzyme regulation. The allosteric regulation, covalent, and different other kind of regulatory strategies you might have already aware of. I'll try to apply those principles in respect to today's topic. That is the regulation of glycogen metabolism, which is a cornerstone topic in biochemistry. That's what the title given by your department. So now let us get into the topic. Regulation of glycogen metabolism. To begin with, glycogen. What is glycogen? Glycogen is a polymer of glucose. It is a homopolymer made up of glucose residues, and which is used as a storage molecule in animals. So what is its counterpart in plants? It is starch. Starch is a storage carbohydrate in plants, while glycogen is a storage molecule in animals, carbohydrates. So glycogen is a huge molecule. It contains almost up to 1 lakh of glucose residues in it, which is an extensively branched molecule that is encircled into like many circles. Like it is branched at almost at every 10th residue. The branching point, you know pretty well, the branching point is made up of alpha-1,6 glycosidic linkages, whereas the regular linkage is alpha-1,4. So this branching occurs at every 10th residue in the outer circles. Coming to the inner circles, that is core of the structure, the branching occurs as frequent as every 4th residue. So that shows how concentratedly organized the structure of glycogen is. Now the question arises, like all of us know pretty well that the storage molecule of human body, the storage of energy or the storage of fuel source is fat, not carbohydrate. That is triacylglycerol is considered as a storage molecule of human body. But now we are talking about glycogen as a storage molecule. So there must be some difference. Going to the preferences through evolution, the fat has been chosen as the storage form of humans because the fat, chemically we call it as acyl glycerols or triacyl glycerol. That is chosen as the storage form because it is very energy rich compared to either carbohydrate or protein. A 1 gram of fat yields almost 9 calories upon burning compared to just 4 calories by carbohydrate or protein. This is because the triacyl glycerols occur in highly reduced form. That means the glycerol molecule is esterified to 3 fatty acids, making it completely reduced, so making it energy rich. At the same time, it is totally anhydrous as well. Anhydrous means totally free from the water. That means the fat can be stored in its pure form, unlike carbohydrate or protein, which cannot be stored in the pure form. Since they are hydrated molecules, they can be stored only in association with water. I'll give you a simple example. If you want to store glycogen, 1 gram of glycogen, you cannot store 1 gram of glycogen as such. You need to add 2 grams of water along with it. That means with the 3 grams of weight is yielding how much energy? Just 4 calories, because there is only 1 molecule of glucose there. 1 gram of glycogen is giving only 4 calories. Suppose if we store the fat, the same weight of fat, that is 3 grams of fat, since it can be stored totally free of water, so how much of energy can be stored with 3 grams of fat? 9 into 3, that is 27 calories. Suppose if the same amount of carbohydrate is stored, how much it is yielding? Only 4 calories. That means a stored fat is yielding almost more than 6 times of energy compared to that of glycogen or protein. That is the reason during evolution, the organisms chose fat as a storage form of energy. Now, suppose if you want to store the same amount of energy that what we are storing, almost 10 to the power of 5 calories, that is 1 lakh calories per adult human. If you want to store the same amount of energy in the form of glycogen, because since glycogen cannot be stored pure, it needs some water. So the body weight will increase enormously. That is the reason fat has been chosen as the storage form of energy, and glycogen or glucose is preferred to yield energy as an instantaneous molecule. Now, a simple question we can put to ourselves, like when fat is chosen as a storage form, we are talking about glycogen as a storage form as well. So what is the difference as a storage molecule? Fat is a long-term storage molecule. Coming to glycogen, glycogen is considered as an instant source of energy. That is, the human brain, which has got an obligatory requirement for glucose. That means human brain can survive only on glucose as the fuel source and nothing else, except in case of emergency starvation. It's only the glucose that is preferred by human brain. That means certain amount of glucose has to be always available in the bloodstream to supply energy for the brain. That means there must be a constant absorption of glucose across the gut walls. That is not possible because we don't eat 24/7. We eat only 2 or 3 times a day. That means in between meals, when, in the post-absorptive, immediately after meal, what happens? The glucose gets absorbed across the gut walls, enter into the blood and available for the entire body, including brain. But between meals, for example, between breakfast and lunch, between lunch and supper, and during the overnight fast, the glucose is not absorbed. It's already absorbed and it's gone. It's utilized. So during this phase, the glucose that is stored in the form of glycogen, especially in the liver, is released into the circulation so that a normal quantity, an adequate quantity of glucose is maintained in the bloodstream, which can be used for the needs of brain and other vital organs. So this is the reason certain amount of glucose is stored in the form of glycogen, the liver and skeletal muscles. This is one aspect. Why some amount of glycogen has to be always stored in liver. This is to maintain a constant supply of glucose to human brain. Number one. Next, glucose is the only molecule that yields energy even in the absence of oxygen. You know about anaerobic glycolysis. That means during strenuous muscular activity, during vigorous muscular activity, when there is shortage of supply of oxygen, still glucose can yield energy through anaerobic glycolysis that you have already learned. So these are two major reasons. To ensure a constant supply of glucose in the blood for the utilization of human brain, and since it acts as an instant source of energy during extensive muscular activity through anaerobic glycolysis. So these are the two major reasons. Glucose has to be stored and available for the function of human body, for the normal function of human body. Now, so with this brief introduction about glycogen, so let us move into the metabolism of glycogen. Coming to the metabolism of glycogen, I think you might have already learned how glucose is stored as glycogen through a process called as glycogenesis, and how the glycogen degrades, breaks down, releasing glucose molecules into the blood through a process called as glycogen breakdown or glycogenolysis. So glycogenesis, glycogenolysis, you might have learned very well. And of course, as I have just mentioned before, all the major metabolic pathways have one or more key enzymes, which are called as rate-limiting enzymes. These two processes, genesis and genolysis, also have rate-limiting enzymes for them. So what is the rate-limiting enzyme for glycogenesis? It is glycogen synthase. Whereas for glycogenolysis, it is glycogen phosphorylase. I hope these two topics are already taught to you. You are very well aware of the various enzymes, especially the role of the so-called rate-limiting enzymes of genesis and genolysis. So our interest is, our topic today will revolve around these two key enzymes of genesis and genolysis, glycogen synthase and glycogen phosphorylase. Am I clear? Right. Now, let us take up how these two processes, glycogenesis and glycogenolysis, are regulated. So these two pathways, since they are diagonally opposite in nature, one stores, other one releases. One stores glucose, other one releases glucose. So they do not occur simultaneously. That means genesis and genolysis do not operate at once, but they do operate on different timings as per the needs of the organism. When the body requires storage, that means when immediately after taking meal, in the post-absorptive phase, when glucose is entering into the blood across the gut walls, there's plentiful of glucose molecules everywhere. So that is the time the glucose is stored. The excess glucose is stored as glycogen in liver as well as muscle. Then in between meals, as I just mentioned, when glucose is not available in the circulation, then glycogen will degrade and release glucose into the stream. That means in the post-absorptive phase, glycogenesis is active, glucose storage is active, and during in between meals time and during the overnight fast, glycogenolysis is active. That means these two processes of genesis and genolysis are regulated reciprocally. They are regulated reciprocally. They do not occur at once. Are you following? Please raise your hands. Yeah, fine. Thank you. I will give you a very simple example how these major metabolic pathways are regulated reciprocally. You must be knowing car driving, or most of you must be knowing bike driving at least. While driving a car or a bike, what we normally do, we operate the accelerator as well as the brake. Do we operate both at once? We don't. When you want to speed up your vehicle, you put your foot on the pedal of accelerator. When you want to slow down or stop, you put your foot on the brake. So while driving a car or a bike, we will be constantly operating the accelerator pedal as well as brake pedal as per the requirement and as per our speed on the road. The similar fashion, all the major metabolic pathways here, the genesis and genolysis, are just like the brake. They will be used as a brake and accelerator in a car. That means when one is up, other one is down. When one is operating active, other one is inactive, inoperating. Okay, so they are operative reciprocally. So they will be regulated reciprocally. Okay, when glycogenesis is up, glycogenolysis is down, and vice versa. Right? Now, so let us come back. So I just mentioned in the beginning of my lecture that the so-called rate-limiting enzymes of these metabolic pathways that regulate the metabolism are finely regulated. They are very finely tuned. They're finely modulated. For that sake, the body has evolved various strategies that you have learned while learning about the regulation of enzymes, the chapter of enzymes. So I don't repeat all that, but I'll try to take up the topic that is required for today's topic. I'll just try to apply those principles of regulatory strategies which you already knew very well, applying to today's topic, that is regulation of glycogen metabolism. Just a moment. All right, fine. Okay. Now, so coming to glycogen metabolism. So we have the body has adopted, the cell has adopted to two types of regulatory mechanisms. The first one is allosteric regulation. The second one is covalent modulation. What are they? Allosteric regulation, covalent modulation. First, let us take up allosteric regulation, later followed by covalent regulation. Allosteric regulation. As we know pretty well, all the enzymes have binding sites, right? All the enzymes have binding sites. When there is a enzyme substrate interaction, various attractive forces, mostly the non-covalent forces, which you are very well aware of. What are those non-covalent forces? Electrostatic interactions, hydrogen bonding, hydrophobic interactions, van der Waals, all these non-covalent forces. So these attractive forces are operative between the substrate and the enzyme. When the substrate approaches the enzyme, these attractive non-covalent forces will change the conformation of the active site on the enzyme. So they make it little bend so that it can accommodate the substrate very well. so that that will completely attach to the enzyme. So we call it as induced fit theory. You might have learned, you just recall your knowledge in enzymes. We learned about induced fit theory. Okay, so the conformational change happening in the active site of enzyme makes the substrate to be properly fit into the enzyme, so that enzyme substrate complex is formed, and that is the beginning step of biocatalysis. Now, so these conformational changes happening in the active site is capable of changing the conformation of the total enzyme. Not only the binding site, the change happening in the binding site, that is active site, also alters the complete conformation of the enzyme. And as you know, well, when learning about enzymes, not all enzymes are alike. There are different types of enzymes. Normal enzymes, isoenzymes are there, allosteric enzymes are there, multimeric enzymes are there, different types of enzymes. So there are certain number of enzymes, some important enzymes, which have an additional binding site apart from the active site. So there are certain other molecules can bind to this additional active site. I'm talking about an additional site, additional binding site, not the regular active site where the substrate binds. So this additional site, so there are certain enzymes that possess additional sites for binding, or these sites are called as allosteric sites, and the molecules that bind to these allosteric sites to influence the enzyme activity are called as allosteric molecules, allosteric effectors, and the enzymes that have these allosteric sites, allosteric binding sites, are called as allosteric enzymes. So here, coming to our glycogen, the glycogen synthase and glycogen phosphorylase, these two are allosteric enzymes. That means they have their allosteric modulators. And allosteric modulators or allosteric effectors are of two types, activators or inhibitors. That means allosteric binding sites occur as two types, activator site, the inhibitor site. When an activator, allosteric activator binds on the allosteric site, that alters the overall conformation of the enzyme. That includes the active site as well. So making the enzyme to, the substrate to fit into more properly, more precisely into the enzyme, so that augmenting the activity of the enzyme, the biocatalysis of the enzyme multitude in more numbers, more actively, more speedily. This is done by the allosteric activator. On the contrary, if the binding molecule is an inhibitor, allosteric inhibitor, that does the opposite. That also makes some conformational change that loosens the enzyme substrate binding in active site, so that the enzyme biocatalysis is, enzyme catalysis or enzyme activity is inhibited. That means, in a nutshell, the so-called allosteric molecules, if it is an activator that increases the activity of the enzyme, conversely, if it is an inhibitor that is binding, that decreases the activity of the enzyme. Hope it is very clear. That means there are certain molecules, certain metabolic intermediates that act as either activator or inhibitor by binding to the allosteric sites of these enzymes. I hope it is very clear to all of you. Now let us apply this to our glycogen metabolism, how glycogen metabolism, the genesis and genolysis are influenced by this allosteric regulation. First, let us take up genesis. When genesis is active, after taking food, that is in the post-absorptive phase, when glucose is plentiful. That means the glucose is polymerized into glycogen. So during that phase, what happened? So what is the most commonly occurring molecule during glycogenesis? It is glucose 6-phosphate. So glucose 6-phosphate occurs in big numbers, large concentration. So glucose 6-phosphate is an allosteric effector. It acts as an allosteric activator for glycogen synthase, which is the rate limiting enzyme of glycogen synthesis. I hope it is very clear. That means in allosteric regulation, glucose 6-phosphate, which is an intermediate of glycogenesis, activates glycogen synthase, the rate limiting enzyme of glycogenesis, and enhances the process of glycogenesis. That means glucose 6-phosphate favors glycogenesis, augments the speed of glycogenesis. I think it's very clear. Conversely, on the other hand, the same molecule of glucose 6-phosphate, along with ATP, of course, during the relatively fasting phase, during overnight fast or between meals, when glucose levels are down, low, the energy state is low, the glycogenolysis has to operate because it has to contribute glucose to the blood, right? Okay. When the post-absorptive phase is over, it's almost 3 hours since you had your last meal. The glucose levels have dwindled down, have come down in the bloodstream, in circulation. Then new glucose molecules have to be added to maintain the normal blood glucose level. For that sake, glycogen that is stored in liver will start breaking down and releasing glucose into circulation, where these two glycogen, these two allosteric regulators, glucose 6-phosphate and ATP, earlier which were favoring glycogenesis, that will favor glycogenolysis now by inhibiting the glycogen synthase. That means the glucose 6-phosphate, the key molecule of glycogen metabolism, is acting as an allosteric activator for glycogenesis and allosteric inhibitor for glycogenolysis. Please, you remember this. So this is briefly about the glycogen synthesis and degradation, how they are regulated by allosteric regulation. Now, let us move on to the second strategy, the second type of regulation. That is, what is the second one? Covalent modulation or covalent modification. You can call anyway. So allosteric regulation is the first regulatory strategy to regulate glycogen metabolism, and covalent modulation is the second regulatory strategy that limit the rate of glycogen metabolism. Now let us see what do you mean by covalent modulation or covalent modification. Covalent, the name itself, denotes to covalent bonds. In contrast to what we have seen in allosteric regulation, where the so-called non-covalent forces are operative. In covalent modification, it is the covalent bonds that are operative. That means the key enzymes, here the glycogen synthase for genesis and glycogen phosphorylase for genolysis. These two are subject to covalent modulation by certain charged chemical groups. The charged chemical group can be anything. Usually it is a phosphate moiety, a phosphate group. The phosphate group will attach to the active site of enzyme, to one of the amino acids. Enzyme is a protein. So naturally, amino acids, the active sites mostly it is the hydroxyl amino acids like the serine, threonine, tyrosine that occur in large numbers. So these charged groups, charged chemical groups, the phosphoryl group here will attach to the amino acids in the active site of the enzyme. That means the enzyme gets phosphorylated. The enzyme gets phosphorylated. So certain enzymes become active when they get phosphorylated. Certain other enzymes will become active upon losing this phosphate moiety. That means a reversible phosphorylation. That is phosphorylation, dephosphorylation. And alternate phosphorylation, dephosphorylation occurring on the same enzyme keeps it either active or inactive, depending on the type of enzyme. I will explain. Not to get confused, I'll explain. In clear about taking the help of some PPTs, I'll explain in respect to these key enzymes. So here, covalent modulation regulates enzyme activity by either adding a phosphate moiety or losing a phosphate moiety. That means either by getting phosphorylation or undergoing dephosphorylation, right? Of course, this phosphorylation and dephosphorylation happens through a array of chemical cascade, events of chemical cascade. That means various chemical signaling events, mostly beginning from the cell membrane into the cytosol. Various signal transducing events, signal transmitting events will determine how the metabolism can be regulated through covalent modification. And of course, please remember that covalent modification is only a means, only a mechanism. The real player is hormone here. So hormones do influence the enzymes, the so-called rate-limiting enzymes, through their action, through the mechanism of covalent modification. So I'll try to explain what does it mean by covalent modification with the help of these PPTs. So shall we please share the PPT number 1. This one. Student, yeah. నేను కనబడుతున్నా మరి ఇది కొంచెం పక్కకే జరిపేసాను ఇటు పక్క ఇటు పక్క రాని జస్ట్ మూమెంట్ ఇంకొంచెం వస్తుందా ఫైన్ ఇట్స్ ఓకే కొంచెం కిందికి ఏమైనా వస్తుంది కొద్దిగా కిందికి రైట్ ఓకే పర్లేదు అది పైకి ఓకే ఫైన్ యా మన బొమ్మ కనబడి సరిపోతుంది పోతుందా మాటి చూద్దామా స్టూడెంట్స్ ఆర్ యూ ఏబుల్ టు సీ ద పీపీటీ ఉంటలేదండి దాంట్లో Are you able to see the PPT? Please raise your hands. Not able to. Okay. Just a moment. Malini, are you on the line?
Yes, Dr.
Sir.
I'm here.
Well, share your number.
Can you click on share button on the bottom?
Share
sir?
button there.
At the bottom of the screen, sir.
Yeah, share button they have clicked. Share.
Then select the entire screen. and click on share.
I think the PDF is making some problem, I think. That is the reason
You-
they're
you-
not able
you
to see
can-
the video.
You can share the
Yeah,
PPT.
yeah,
sir.
fine.
Yeah.
ఇప్పుడు ఇక్కడ ఇది షేర్ చేయండి చూద్దాం.
From beginning.
Video
Can click
కనబడుతుందా?
on this.
Can it
Are you
be,
able to see the diagram?
can it be zoomed, sir?
Zoom చేయండి. Zoom.
Also, he needs to go to the slide show option. You're not presenting as yet. You're still in the editing mode.
That's okay. I think the center part, sir, you zoom it. We can just continue this way.
ఇది ఫస్ట్ ఫిగర్. Zoom కాకపోతే వాళ్ళని చేయమన్నాం. ఇది కాదబ్బా. ఫస్ట్
Can you click on from beginning button?
ఇది చేయి.
Yeah.
ఇది జూమ్ చేయగలమా?
Click on that button, sir.
Button click.
They're clicking, but it's not
Clicking,
working.
there is going to the next one. Okay, Malini ji.
Yes, sir.
Hello, Malini. I
Yes.
think these people are not able to do that. Can you please
Can you-
share
Sure.
from your side?
Sure. I'll share it. You'll have to stop sharing.
Stop this.
You'll have to stop sharing for me to start sharing.
Stop share the Jesse.
I'll stop.
Still,
no problem.
look, yeah.
Yeah, I stopped Malini. You can do that.
Now you people you can just share the slide one. You can share, Malini.
Yeah.
వస్తుందా అక్కడ? వస్తుందా? వాయిస్ క్లియర్ ఉంది కదా? వీడియో అంతా రైట్, ఓకే. Yeah, Malini, please share the upper half of the first slide. Yeah, please try to move your cursor, the upper half.
Just 1 second.
Malini, I think you can stop. Yeah.
Yeah, fine. Is it okay, students?
Some more, sir. Malini, some more. Yeah. Fine?
Better?
Reduce little bit. You have to move towards a little right. Because the student, yeah. Little more right. Little more. Little more.
Oh, 1 second. It.
Yeah, I think it should be fine. Now it's fine.
Okay.
It's good. Please don't move it anymore.
Okay.
It's fine.
Okay.
The students, are you able to see the PPT? Is it visible to you? Please raise your hand. Yeah, fine. Thank you. So, okay, let me resume. Let me resume back. Yeah, please, the begin here. There's a green colored box. I'm showing begin here. Okay. So underneath the coil one, that shows the plasma membrane. Okay. Now I'm telling you that it is the hormones that regulate these metabolisms through covalent modulation. Okay. So here the hormones are glucagon and epinephrine on one side and insulin on the other side. Okay. They exert opposite actions. So glucagon and epinephrine, which are peptide hormones, okay, being water soluble, they'll reach out to every cell of the body, of course, their target organs in the free form because of their hydrophilic nature, where they, upon reaching the target organ cell membrane.
1 second, sir. 1 second, sir. 1 second.
Yeah, sure.
Yes. Continue, sir. Continue.
Can
So
I?
sorry.
Yeah,
Yes.
fine. So these two hormones, glucagon and epinephrine, will reach out to their target organs, target cells, where they have to exert their function. They need to enter into the cell, but they cannot do so because of their polar nature, because of their water solubility, because of their hydrophilic nature. Since the biological membranes are predominantly hydrophobic, they cannot traverse across the plasma membrane. Hence, they bind to one of the receptors in the plasma membrane, forming HR complex that I am showing here, the red one. The red one, that is HR complex. HR complex stands for hormone receptor complex. Okay. So when the hormone is binding to the receptor, forming HR complex in the plasma membrane, that stimulate a special protein that occurs in these cells called as G protein. I'm showing it here, just the down G protein. That is a trimer. Trimer means 3 polypeptides, 3 subunits, alpha, beta, gamma. They are shown in 3 different colors: light green color, saffron color, and light blue color, alpha, beta, gamma. And it is shown GDP, the guanosine diphosphate, GDP attached to alpha, right? So this is called as G protein because it is dependent on guanine nucleotide. So this G protein is a trimer, is a heterotrimer because it has different types of subunits, alpha, beta, gamma, bound to GDP. So it is said to be inactive. It is said to be in the sleep mode or off mode, off position. So the formation of HR complex, that is the hormone binding to the receptor, stimulates this sleeping G protein to wake up it, to make it active. That means the GDP is replaced by GTP. The GDP is replaced by GTP. That means there is a nucleotide exchange of GTP to GDP. That means, and the trimeric molecule will dissociate into 2 fragments. Alpha fragment with the GTP on one side, beta and gamma on the other side. Okay. So among these 2 fragments, the alpha component with the GTP is active one. So this is the active form of G protein. Okay. So this active form of G protein immediately stimulates a very important enzyme occurring in membranes called as adenylyl cyclase. I think you are able to see adenylyl cyclase. So this enzyme adenylyl cyclase is activated by the active G protein. Okay. Once it activates adenylyl cyclase, what happens? The GTP loses a phosphate, becoming GDP. That becomes inactive. So this GDP alpha will combine with beta alpha to regenerate the original molecule. That's fine. So that means the alpha with GTP has got a very short half-life. Its biological half-life is very, very short because this active G protein has got an intrinsic GTPase activity. That means an intrinsic within inbuilt GTP hydrolyzing enzyme, GTPase. As soon as it activates adenylyl cyclase, that it gets hydrolyzed and become inactive. The inactive molecule will again stimulated by HR complex and recycle again and again and again. So this is a cyclic process. I think you understood. That means the hormone is able to stimulate G protein. The stimulated G protein further activated an important enzyme, adenylyl cyclase. Now, the adenylyl cyclase, the activated adenylyl cyclase will convert ATP into cyclic AMP. So this adenylyl cyclase converts adenosine triphosphate into cyclic AMP. As all of you know pretty well that cyclic AMP is a wonderful molecule because it acts as a messenger, a signal transducer, a signal transmitter. That means the original message that is carried by the hormone, whether it is epinephrine, norepinephrine, or glucagon, the same message is carried by cyclic AMP here. That it is not the hormone that is directly exerting its action, but it is the cyclic AMP acting on behalf of the hormone. That is the reason cyclic AMP is called as second messenger. Cyclic AMP is called as second messenger. So what is the first messenger here? The hormone itself. Hormone itself is the first messenger. So it is the second messenger. So all these hormones, all these hydrophilic hormones, which cannot enter into the cells, do not function directly, but they do function. They do act through their respective coenzymes. So cyclic AMP is one of the very, very important second messengers. Now, the cyclic AMP. Cyclic AMP in turn stimulates one more enzyme called as protein kinase. It in turn stimulates one more enzyme called as protein kinase. So protein kinase is a tetramer. Tetramer means 4 subunits, which has got 4 subunits, 4 polypeptides. 2 regulatory polypeptides, 2 regulatory subunits, 2 catalytic subunits that are shown as R, C, and C. Okay, so protein kinase that is stimulated by cyclic AMP is a tetramer, is a heterotetramer having 2 regulatory subunits and 2 catalytic subunits. So 4 molecules of cyclic AMP will bind to the binding sites on regulatory units of protein kinase A. So this enzyme is protein kinase. A is because it is activated by cyclic AMP. Okay, so protein kinase. So when cyclic AMP binds to protein kinase, that gets activated. That means the protein kinase dissociate into a regulatory unit and catalytic unit. The catalytic unit, the C and C, which in turn phosphorylate the enzyme, a protein or enzyme, converting it into a phosphoenzyme, a phosphoprotein or phosphorylated enzyme. Are you able to? That means the protein kinase, there is only one function of protein kinase or kinase. Kinase enzymes are always add a phosphate group. Okay, so here the protein kinase is adding a phosphate group to the enzyme, converting it into a phosphorylated enzyme. So this phosphorylated enzyme will elicit the final biochemical response in the cell. Okay, that means we have seen a array, a line of signal transducing molecules, right from the hormone receptor to hormone receptor to G proteins to adenylyl cyclase to cyclic AMP to protein kinase to phosphorylated enzyme phosphorylation. So these are the various molecules that happen the changes happen one by one, finally eliciting the ultimate biochemical response or physiological response. That is the reason I have called initially, if you remember, this entire sequence of events as signal transduction cascade. I called it as signal transduction cascade. That means signal transmitting events in a sequence. Okay, so signal transduction cascade, the signal transmitting events in a sequence, happening in a sequence, right from the hormone binding to its receptor to the ultimate biochemical response by the phosphorylated enzyme, right? Okay, so this is in principle about how the hormones elicit their function through their second messengers through covalent modification. Okay, so in covalent modification, these enzymes, the last part of the diagram, the enzyme gets phosphorylated. Okay, just please show this once again. The same figure, the bottom one, please. Yeah, please. Right. If you observe here, the enzyme is phosphorylating by kinase enzyme. Conversely, the phosphorylated enzyme can lose phosphate by another enzyme called as phosphatase, protein phosphatase. I have shown protein phosphatase. That means the phosphorylated enzyme become dephosphorylated enzyme. If you remember, I told you that certain rate limiting enzymes are active in phosphorylated form. Certain other enzymes are active in dephosphorylated form, right? and only little up, please. And you please observe one more site here. If you observe the ATP to cyclic AMP, okay, if you observe cyclic AMPs can be converted to 5' AMP. That means cyclic AMP is a second messenger, active. This simple AMP is not active, inactive. So there is enzymes called as phosphodiesterase. Phosphodiesterase that converts cyclic AMP to 5' AMP. That means active cyclic AMP to inactive one, inactive molecule. That means if you recall once, the original hormones, glucagon and epinephrine, are phosphorylating the enzyme. Okay? And on the contrary, insulin, which has a counteraction to glucagon and epinephrine, will stimulate, induce, or activate phosphodiesterase enzyme so that cyclic AMP will be inactivated. Okay? Please come down. On the same lines, then the hormone, little more down, the hormone insulin also activates enzyme protein phosphatase. That means that makes the phosphorylated enzyme into dephosphorylated one. Right. So in a nutshell, enzymes phosphorylated and dephosphorylated. That means every enzyme, every rate limiting enzyme occurs in two forms. One phosphorylated form, other one dephosphorylated form. That means active form, inactive form, or on form, off form. Like you can compare this with a electric switch. We use the same switch to put on a fan or put off the fan, to put on a light or put off the light. So it occurs in two states. The same manner, these enzymes have an on-off states. Okay, phosphorylated, dephosphorylated. Now, so let us apply this principle, okay, this concept to our glycogen metabolism. Please, next slide, please. Yes, now let us take up glycogenesis. We can go a little faster now as time is approaching. So you please observe this diagram. Same thing, what you have just learned, glucagon, epinephrine. Okay, they are stimulating G proteins. Are you able to see? They are stimulating G proteins. The G protein is activating adenylyl cyclase. We have just learned. The activated adenylyl cyclase is converting ATP into cyclic AMP. The cyclic AMP is activating protein kinase, cyclic AMP dependent protein kinase. Okay, so the cyclic AMP dependent protein kinase, upon activation, what is doing? It's phosphorylating glycogen synthase, which was earlier in dephosphorylated form. It is phosphorylating glycogen synthase. So please observe here. The glycogen synthase is active in dephosphorylated form. It gets inactive when gets phosphorylated, upon phosphorylation. Okay, that means these two hormones, glucagon and epinephrine, inactivate glycogen synthase. Glucagon and epinephrine hormones inactivate glycogen synthase by phosphorylating it. That means these two hormones inhibit glycogen synthesis. These two hormones inhibit the glycogen synthesis. That is what I have shown here in the pink colored arrow here. On the contrary, insulin, by activating phosphatase enzyme or inactivating cyclic AMP, it favors glycogenesis. I think you got the point. I repeat, not to get confused. It's very simple. Very, very simple. Glycogen synthase occurs in two forms, active, inactive. Active is dephosphorylated form, inactive is phosphorylated form. Right? Okay. So, hormones, epinephrine and glucagon, inactivate glycogen synthase, thus inhibit glycogen synthesis, whereas insulin keeps the enzyme glycogen synthase as in the dephosphorylated form so that glycogenesis is favored, or by inhibiting the synthesis of cyclic AMP itself, insulin, through phosphodiesterase. In that way also, the two-level regulation, insulin, favors glycogenesis. So in a nutshell, insulin favors glycogenesis, and the counter-insulin hormones like glucagon, epinephrine, norepinephrine will inhibit glycogenesis. Now, I hope you understood. Now let us move to the next slide. That is where let us take up genolysis. We have just seen how glycogenesis is regulated through covalent modification, through hormones, and how glycogenolysis gets affected by the same hormones, glucagon and epinephrine. As you know, they are reciprocally regulated. You can easily say that they'll activate. Now, so same glucagon and epinephrine, they'll activate G proteins. Activated G protein will activate adenylated cyclase. Same. The mechanism is same until the end. Okay? The same mechanism. Activated adenylated cyclase convert ATP to cyclic AMP. So, of course, there's a typing error here. It is not cyclic ACP, cyclic AMP. It's a typing error. Cyclic AMP, right? Okay. So, cyclic AMP will activate protein kinase. So, what is the function of protein kinase? Phosphorylating the enzyme. Here, it doesn't directly phosphorylate glycogen phosphorylase, the rate-limiting enzyme of glycogenolysis, but it phosphorylates another enzyme called as phosphorylase kinase. Phosphorylase kinase. Okay, it phosphorylates. So, the phosphorylated phosphorylase kinase becomes active. That further phosphorylate glycogen phosphorylase, the original enzyme of genolysis. So, glycogen phosphorylase, the rate-limiting enzyme of glycogenolysis, upon phosphorylation gets activated, favoring glycogenolysis. Conversely, insulin, by dephosphorylating the glycogen phosphorylase, inhibit glycogenolysis. Okay, here insulin is shown at 3 places. If you observe the figure, it is given in the book also. You can observe later on. You can just go through at leisure. Right. So that means, okay. So I'm almost coming to the end point of the lecture. To summarize, the hormones, glucagon, epinephrine, norepinephrine, will inhibit glycogenesis and stimulate or enhance glycogenolysis. On the contrary, conversely, insulin favors glycogenesis and inhibit genolysis. That means, so this is how these different hormones, insulin and counter-insulin hormones, will reciprocally regulate genesis and genolysis as per the needs of the cell, as per the needs of the tissue, as per the needs of the organism as a whole. So when the glycogen has to be stored, glycogenesis is favored in the post-absorptive phase. When glycogen has to break down between meals and during overnight fast, glycogenolysis is favored. So this entire thing is mediated by hormonal action, which in turn exert their action through the respective second messengers. The second messenger here is cyclic AMP. Okay, so this mechanism is called as covalent modification or covalent modulation. Okay, anyhow, dear students, you might have learned the very basics of this covalent modification allosteric regulation while learning regulation of enzymes. Now your teachers, of course, I taught you how they can be regulated in respect to glycogen metabolism. You learned it in detail once again in hormone action. Now, so let me conclude in a couple of minutes, applying the same to other metabolisms as well. Of course, you might not have completed other metabolisms. I'll just briefly summarize that. This is very, very important topic, because if you learn this topic, you'll be able to answer the regulation of all major metabolisms, whether it is a carbohydrate, lipid, or amino acids or nucleotides. You can answer any question on regulation of any of the rate limiting enzyme of any of the major metabolic pathway. Mechanism is the same. We have seen how glycogen synthase and glycogen phosphorylase have regulated glycogenesis and glycogenolysis. So where glycogen synthase is active in dephosphorylated form, glycogen phosphorylase is active in phosphorylated form. Let us apply to other metabolisms, like which you have already finished. Like for example, let us take up some enzyme in fatty acid biosynthesis. Or okay, let us take up the simplest example like PDH. You might have already studied PDH that converts pyruvate to acetyl-CoA. The PDH is also active in dephosphorylated form. Glycogen synthase is active in dephosphorylated form. Glycogen phosphorylase is active in phosphorylated form. Coming to lipid metabolism, fatty acid biosynthesis. The rate limiting enzyme is acetyl-CoA carboxylase. That is active in dephosphorylated form. That means inactive in phosphorylated form. The rate limiting enzyme of cholesterol biosynthesis. What is that? HMG-CoA reductase. That is also active in dephosphorylated form. Inactive in phosphorylated form. The hormone sensitive lipase, the rate limiting enzyme of lipolysis, the breakdown of lipids, acylglycerols, is active in phosphorylated form. So these are the few examples. These are the few examples. And not to get confused, here all the regulatory enzymes or rate limiting enzymes are, some of them are active in dephosphorylated form. Some of them are active in phosphorylated form. I'll give you a simple hint, a simple secret, how to remember them very easily. Please remember, the enzymes, the are active in dephosphorylated form, or the enzymes that are associated with synthetic processes. That means when a synthesis is happening, like glycogenesis, glycogen synthesis, okay, fatty acid synthesis, cholesterol synthesis, all these are active in dephosphorylated form. And the enzymes associated with degradation, breakdown, lysis are active in phosphorylated form. Lipolysis, glycogenolysis, right, are active in phosphorylated form. This is the easiest way to remember, not to by heart. It's very, very simple. So that means please remember finally that the so-called rate limiting enzymes of all the major metabolic pathways occur in two states, active, inactive, that is either phosphorylated or dephosphorylated. Okay, I hope with this 90 minutes lecture, I'm able to make you understand the regulatory strategies of metabolic pathways, especially in respect to glycogen metabolism. And I suggest to all of you to please go through the textbook once, this topic. You can recapitulate once again in the enzymes chapter by reading Covalent modification allosteric regulation once there, and glycogen metabolism. And if time permits, read the hormone action, chapter number 40. Hormone action under group 2 hormones. You will understand everything. You can master this topic, which is common for all the regulatory strategies of all the major metabolisms. Yes. Hope you enjoyed this lecture, my dear students, both at Somerville Memorial College and Surabhi Medical College, my students. I hope you enjoyed the lecture. Please raise your hands. Thank you very much. Thank you very much. And I conclude my lecture here with a note of thanks to the respected HOD and my good friend of mine, Dr. Vijayta Thomas, and the principal of Somerville Memorial Medical College, CSI Medical College, and all the students, the faculty of Department of Biochemistry for attending this session and listening to me very attentively. I thank you one and all. Thank you.
Thank you. Thank you so much, sir.
Now, if time permits, madam, shall we give chance to students to put any queries
Sure, sir.
for a few minutes?
Any doubts? Any questions? Anywhere? Sir has touched all aspects, even enzymes. He has touched on all. So anything, you have any doubts, anything you can clarify with, sir. Anyone? Any doubt?
Any doubt? If you need more clarification, more clarity on any of the covered topics, anywhere, if you have any doubt, you can straight away put me a question. I'll love to answer.
Yes, anyone? Anything from, you want again enzyme, even regulation, sir has touched so beautifully, gone from basics. So anyone need anything, little bit more clarification on anything, regulation, anywhere, glycogen, just please free to ask.
So I'm happy that they understood it so well.
You have taken it so simplified and, beautiful way. So starting from basics, touching on the regulation, brushing up their basics from there, ours too. So I'm sure all of you enjoyed the session. So 2 words from student, right? So our students would like to, say a few words.
Yeah, sure.
So on behalf of the 24 batch, I would like to extend my heartfelt thanks for this 2-hour session. Through your efforts, sir, we were able to learn quite a bit today. And from the bottom of our hearts, we'd like to thank you so much.
Morning, sir. So we would again like to give my heartfelt gratitude to you and all 150 of us. Really liked the session. It was really nice that you started from the basics and clear every single concept without any doubts. Thank you so much, sir, for involving us, coming to spend time with us in your busy schedule. Thank you so much.
So thank you, sir, once again for a smiling, beautiful session, Calmly you have taken with a beautiful smile on your face. I'm sure all of you enjoyed it and loved it. Thank you so much.
Thank you very much. I hope to see you all once again some other time.
So we would now love to have you here. Next time we'll make it, you physically here, not virtually.
Allowed to be there. I'll try to make a trip.
And, so we'll be taking feedback and we'll definitely share with you the feedback also.
Fine. Thank you, Dr. Vijita. Especially my special thanks goes to you and your dean for giving this opportunity. And I enjoyed the wonderful company of the students of 24 batch, 2K24 of CSI Medical College. Thanks a lot. See you all. Bye.